Matthew to introduce the event.
Well, good evening, everyone. This is my favourite type of school when we're not in the classroom. We are actually talking about relevant 21st century learning and the outcomes that it can bring. We talk about jobs in 5 years, half of them won't be around that we see today. So what will the new jobs be? Well, I think this DNA talk tonight will be just part of that whole experience. You know, living in our COVID world today and what we've learned about, you know, all the sequencing that goes on with genes and what DNA can do. That's just one part of the equation. But what about all the advances it can do to help us in our world, to sustain our world, you know, sustaining our food lines, looking at health, looking at how we can look after our environment. This is an amazing set of technology that's going to be discussed tonight. So again, it's a warm welcome to the classroom of the future, not in four walls, not in a school. We are talking to real live experts. We are talking about the reality of what future jobs are doing now, not just in 5 years' time. And so I hope all of you students will pay very close attention to what might be part of your own life journey in education. The sciences today integrate, and I'm sure that'll be spoken about. So I thank our presenters tonight. It is just so fantastic to have people from the university and students and professors, and also led by our own Miss Vanita and her team. It is an exciting way of schooling. So I'm looking forward to the lesson that lies ahead. So welcome to everyone tonight and literally enjoy, and dare I say, learn. Thank you.
Thank you, Mr. Matthew, for your kind words of encouragement. New technologies have shaped our lives and the physical landscape of our habitats throughout human history. But modern science is now playing at much finer and deeper micro levels to change the architect of the organismic life on the biological landscape. To explore these molecular manipulations, we have with us field we call genome editing. Dr. Mappa is an associate professor at Shivnada University. She received her PhD in biochemistry from Ludwig Maximilian University, Munich in the year 2009. She received the Innovative Biologist Award IYBA from the Department of Biotechnology, Government of India. Um, from the Department of Biotechnology, Government of India, and worked as an IYBA Fellow in the CSIR Institute of Genomics and Integrative Biology, Delhi, before joining Shivnada University. But first, I would like to invite Ms. Sandhya Kasi, a third-year biotechnology student at Shivnada University, to walk us through a fun session to understand with her what genome editing really is.
Hi, thank you for that. One moment, I'll just share my screen. Hello, hi. I hope everyone can see my screen.
Yes, Sandhya, go ahead.
Hi, good evening to everyone. I am Sandhya and I'm a third year biotech major from Shivnada University. And today I'm going to be talking to you about a really up and coming topic, like Mr. Matthew said, that is going to be an integral part of the future. It's about genome editing. So let's just dive right into it. So let's start with the very basics. So what are genes and what do they do? We all have heard this word, and I'm pretty sure all of us have used it as well, whether it's in terms of jokes saying, you know what, you're like your parents, you share their genes, or in different contexts. And what exactly is a gene? A gene is a segment of DNA. And DNA is this double helix X-shaped structure that you see here, which you would have seen very commonly around you in pretty much every poster that has anything to do with biology. And this X-shaped ladder is called DNA. The ladder is formed by bonding between the purple strand and the pink strand that you see here. And the building blocks of DNA are four bases, which I'm sure you all have heard of: A, T, G, and C. There is a lot of chemistry involved in how they bond and the structure of DNA, which we will not get into right now. But yeah, so genes code for proteins, and when I say code, I mean that they dictate how a protein is formed. To understand this better, think of it in terms of a dish and a recipe. A recipe encodes a dish. A recipe decides how a dish is going to turn out. In a very similar way, a gene decides how a protein is going to turn out. Proteins carry out every single function in the cell. And I've come to realize that this concept is not one that's actually very easy to understand. I'm sure that a lot of you, when I said protein, the first thing that came to your mind was probably, do you mean an egg? And that's a very common thing. But when I say protein, I mean proteins in the cell that carry out all functions. For instance, some that you might have heard of are hemoglobin that carry oxygen to different parts of the body and that's what makes your blood red, or chlorophyll, that's also a protein present in plants and that makes the plants green. Or in times of COVID, I'm sure that everyone has heard of antibodies, and these are proteins too. So, like I said, so genes encode proteins, and proteins carry out all functions of the cell. So, to give you a better understanding of how extensive the control of genes actually extends, here's a short video.
Let's start by talking about genes. These are sections of the long DNA molecules coiled up inside each cell of every living thing, from microorganisms and insects to plants and animals, including humans. Think of DNA as the instruction manual for an organism. In people, genes can influence characteristics such as your height and the color of your eyes.
So this is just one small aspect of it that genes control. And like I said, they control everything because they encode proteins. Everything that a cell does, from growing to dividing to shrinking to dying to even just moving, is controlled by cells, by proteins, I'm sorry. So once in a while, there's a mistake in the gene. You have genes that don't encode for a protein that's exactly right. So look at these two sequences here. These two sequences are identical except for a single alphabet. And this causes a major difference in how the protein ends up looking. So this single base change, which results in the difference in how the protein looks, is known as the mutation. Mutations occur naturally in the cell, in that every single time your cell divides, there is a mutation. But more often than not, these mutations don't actually have a physical difference, so you don't notice it. That's probably why you don't see people changing their hair color. They don't spontaneously change hair color, do they? So also editing genes can be done artificially, and we would do this if we wanted to change the protein. Say, for instance, you found out that if you could modify the way this protein looks, it would become more efficient. Hemoglobin could carry more oxygen if you modified how it looks. You'd want to change that, wouldn't you? So you would change, what would you change? You'd change the source that encodes it. That's the DNA. So one single base pair change here, that's from C to T, like I said, the building blocks of your DNA. One single change can make this reasonably squiggly looking protein look like this pretty haphazard structure. The main point to note here is that these two proteins will probably not perform the same function. This can lead to visible differences in how the cell functions and maybe even something more drastic, like in a lot of diseases that we see. So while mutations occur spontaneously, like I said, every time the cell divides is a mutation, we can also induce mutations. If we wanted to change how the protein looked, we can choose to change the gene. So as soon as humans found out that gene gives protein, and editing a gene actually results in the change in the protein, and they realized how straightforward it was, we had to interfere. So we decided to figure out how to edit genes. So there are different ways to do it. One is by selective breeding. This is not so much editing genes as really just focusing on the genes that you want. So to look at a simple example here, you have 4 plants. And say, for instance, you realize that the tall red plants are the ones you want more. You would just breed that over and over again until you phased out the yellow plants. Like you see in this, the third row has only red plants and no yellow plants. Another way to edit genes that in an artificial manner is through radiation. Now, radiations are simply high energy waves. So X-rays are radiations, and UV rays that come from the sun, which the ozone layer protects us from, are also radiations. So radiation is a very simple way to modify DNA. The catch here is that you don't know where the modification is happening. Think of it like this. A cell under radiation is under stress. So for instance, if I gave you 30 seconds and said, write down 10 words that are 10 letters, 10 letter long 10 words, how would you do it? You'd be stressed, right? Your mind would focus on actually writing down 10 words more than it will on spelling them right. In the same way, when your cell is under stress, it focuses on copying the DNA more than it does on copying it right. It has a survival instinct. It says, you know what, I don't care. I can make a few mistakes. I really just need to live right now. That's exactly like you don't know, you can't predict where the changes in the DNA are going to occur. Just like you can't predict which of the 10 words you're going to misspell and how. Another way to modify DNA artificially is using genome editing tools. And we're going to get into this in quite a bit of detail in the future. So I will leave it for then. This is a lot more accurate way of doing it. So can you choose where the changes take place? And yeah, you can. Like I just said, genome editing tools are used to edit genes in particular targeted areas. Because if I study it and I say, you know, if I change this base from T to A, this is a change in the protein, and this is a change I want, then I can choose to do it. Look at this sentence, for instance. The cat ate the rat. If I change the R to an H, you get the cat ate a hat. That doesn't make much sense. Look at it this way. If I change the C to an H, I get the hat ate the rat. And that definitely doesn't make sense. One single alphabet change can have a large impact on a sentence. Look at it in a larger scale in terms of a genome. One single base pair can change a lot. So like I said, genome editing tools allow you to target a particular section of your DNA to cut or to change. Now, how does this change take place? The first step is obviously to cut your DNA. And one common method of cutting DNA is through restriction enzymes. So enzymes are basically proteins that are part of a reaction. They usually help to speed up a reaction. A restriction enzyme is one that cuts DNA. And the important and great part of this is that a restriction enzyme recognizes a particular sequence and cuts just that. So the sequence is usually a palindrome. A palindrome is a word that reads the same in either direction. We've come across a lot of palindromes in our day-to-day life. For instance, mom, dad, those are simple ones, but words like Malayalam are palindromes. They read the same in either way. And this is beneficial in terms of DNA because if you look at it here, whether I read the top strand or the bottom strand, I'm reading GAATTC. So this enzyme, which in this case is called EcoR1, you have a whole lot of them. EcoR1 recognizes this particular sequence. Whichever sample it might be in, whichever DNA from whichever organism, it is going to recognize the same sequence. But if I have a different sequence that I want to recognize, I'm going to have to use a different enzyme. And this is why restriction enzymes are not a common solution for modifying genes of all organisms, because I'll need different restriction enzymes. And when we look at the next slide, we'll understand why having a single system that can be applied to various organisms is actually very important. So restriction enzymes cut DNA, and this gives you ends into which you can insert any other DNA, or you can replace, like here they've replaced the blue strand with a pink strand. So you can do a lot. This cutting the DNA is the first step, which gives you a lot of possibilities. Next, we have CRISPR. CRISPR is a term that a lot of people is being used a lot nowadays. It has become very well known also because it won the chemistry Nobel Prize. Scientists who worked on CRISPR won the chemistry Nobel Prize. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And yeah, it's a mouthful, and which is why we call it CRISPR. So CRISPR was actually originally found in bacteria, which is used as where bacteria used it as an immune system to fight viruses. And scientists working on it looked at it and said, hey, this is a system that we can really use. It's something that can be applied, like I said, across all organisms, and you will understand why. So CRISPR as a system consists of two components. The first is the Cas9 protein, and the second is a sequencer sequence here known as your guide RNA. So think of it in terms of the Cas9 protein being a policeman, and this whole DNA sequence here being a bunch of, say, good guys. But the policeman knows that there's one bad guy hiding in there, and he has this photograph, and the photograph is this green sequence that you see here. So if you were in this place, if you had to catch an odd one out in a sequence, what would you do? If you knew how it looked, you would just simply scan your available sequences, and when you find a sample that matches the sequence that you have, you cut it, which is exactly what the Cas9 protein does. It scans the DNA. After identifying a match, it cuts it. And this itself comes a great way. So just a small point to note here is that, say, for instance, this segment is bad because of a single base, that's the G. So getting back to it, the Cas9 protein cuts out the sequence. And this itself is a really great deal. The ability to be able to find a particular sequence within a large scale is pretty hard. But Cas9 goes another step. It goes a step ahead. It can replace the faulty sequence with a sequence that's right. So the G became the A, and now you have a sequence that makes sense and that's correct. See, look at this in terms of a disease that's caused by a single base pair change, like I talked about. Being able to replace a faulty segment with a segment that's right means that you've cured the disease. And this is just the beginning of the applications that CRISPR has. So before I move forward, I have really covered just the introduction of what genome editing is as a concept and how it can be done. But before I move forward into the applications and how far we've come in this field, are there any questions?
The questions has to be taken from YouTube. So somebody is monitoring, or Mala and Ruchi ma'am, please have a look on that.
Yes, there was one question. A lot of kids are asking what an enzyme is, if you can explain it again.
Sure. So an enzyme is also a protein. And it basically carries out a reaction. So in this case, a restriction enzyme would cut the DNA. So this is just one thing that an enzyme can do. And an enzyme is basically a protein that carries out a reaction. It helps the reaction be carried out faster. So it has a whole bunch of things. Enzymes, you can see them in your day-to-day life, like in your body also. So you have enzymes in your saliva that help in digestion. You have digestive enzymes that help break down the food. So enzymes essentially carry out any process that needs to take place. And they are, like I said, they're protein molecules. So again, proteins do pretty much everything. Anything else?
You can go ahead.
Okay. So moving ahead into the applications and how far we've come with the technology of this sort, let's first look at what we have done in the past, right? So we all have seen examples of you take a white rose and you take a red rose, and if you graft it together, you get a pink rose, changing the color of flowers, crossbreeding you get results like pomato. Pomato is a potato plus a tomato. You get a plant here, like you can see, a plant which has different fruits on the same plant. These are things that we've done in the past, that we've done with traditional methods of plant breeding and things like that. But the thing I'd like to emphasize is that genome editing takes it a step ahead. Genome editing, like I said, unlike radiation, genome editing gives you the specificity of choosing where you want to make the change, and that's why it's different. So obviously, this has taken us farther ahead than these conventional methods have. So let's just look at the short video to see the kinds of things I'm talking about.
Gene editing could make radical improvements to human health. For example, in 2015, doctors used genome editing techniques to cure a baby girl's leukemia. In the future, we may be able to fix the gene mutations that predispose some people to cancer, or enable new therapies for HIV, or edit the genes that cause hereditary diseases. In the plant world, it could make crops more nutritious, disease-resistant, and able to grow in difficult conditions. By editing animals' genes, we could help them resist diseases. Scientists are currently working to edit the genes of mosquitoes to prevent them from carrying malaria. So genome editing has enormous positive potential.
So this video pretty much summed it all up. Genome editing, the things that you can do with genome editing are so much more than we could have thought, say 20, 30 years ago for sure. The idea of being able to genetically modify a pest so that it's not as infective and that could affect a disease like malaria, which causes so much death. All these things that are mentioned in this video are a great leap ahead, really. So let's discuss a few of them, and let's start off with some fun stuff. So things that glow. This is a pretty colorful slide. So using genome editing, scientists have modified organisms so that they can glow in the dark. And how is genome editing used? Basically, it's that we all know jellyfish, and jellyfish glow. So they live in depths of the ocean where there is virtually no sunlight. So they develop the ability for bioluminescence, which is basically that they create their own light. I mean, we've all seen Finding Nemo, the huge puff of fish with the bulb hanging there. That's bioluminescence. It serves as its own source of light. So what scientists did is that they identified the gene that encodes this bioluminescence, and they've put it into different organisms. So here you can see that so this fluorescent rabbit, for instance, you have the genome of the jellyfish, and you have one tiny segment. So say this off, for instance, this off segment that codes for the fluorescing property of jellyfish. And you have the normal genome of the rabbit. And you put the two together and you get a genome which has all the genome of a rabbit and just that one gene from a jellyfish. So it's going to be an ordinary rabbit, but just the ability to glow in the dark. And this is really cool. Also, it's the discovery of the fluorescent protein in jellyfish is very, very important because it has given us the ability to do a lot of things in scientific research that were not possible earlier. Say for instance, these are cells that have fluorescent proteins that are expressing fluorescent proteins. So like I showed in the slide about restriction enzymes, you can use restriction enzymes. I said you can put any sequence between the two cuts that are made by the restriction enzymes. And that's what people do. You put the GFP protein between those cuts and express it in different cells. And this is huge. This is very useful because this helps you track the progress, right? Say, for instance, as an experiment, you have decided to inject a cell with a protein and you want to know, has the protein gone in? There's no way to know. So what you do is you fuse the protein with this fluorescent protein. And when you put it under light, a particular light, you'll see if the cell has fluorescence. If the cell has fluorescence, that means your protein that you inserted has gone in. And that's just one of the many things that GFP is used for in research. This is a more easy to understand example where, say for instance, you're probably tracking movement of a particular protein in the leaf itself. So if you fuse the protein to your GFP, then you'll see wherever the protein goes, the GFP goes. And GFP is very easily observable. So this is just one thing. Beyond this, you have, so also this has been applied in the plant kingdom. So that's pest-resistant crops. Pest-resistant crops are very important. We know that there was a recent locust swarm and things like that. The pests are not going anywhere. So we'll always need pest-resistant crops. So there are examples like cotton, brinjal, and corn. And this might seem like, yeah, sure. I mean, they're doing it somewhere. Somewhere people are having pest-resistant crops and they're sure being used. But what do I have to do with this? Well, it turns out these are very common. Most brinjal that we eat is BT brinjal. Most corn that we eat is BT corn. And most of the cotton clothes that we wear right now are probably from BT cotton as well. So BT here stands for Bacillus thuringiensis, which is basically a bacteria that produces a toxin that can kill pests. So just like they did with the fluorescence, they took this particular gene that produces the protein, they took it from the bacteria and put it in the plant. So now you have a plant that produces this toxin, so it's resistant to pests. Also, this is a very interesting example of pretty much the same thing. It's called scorpion cabbage, and really I was just intrigued by the name. So, so they've taken a very dangerous scorpion that's Androctonus australis, which is the most venomous scorpion in the world, and they've infused the gene of that venom into the cabbage. But now what happens is a gene can behave differently when it's expressed in different organisms. So when this gene, they realized that when it was expressed in cabbage, it was not toxic to humans anymore. Well, this might have been slightly lucky, but that's how it changed. And now they have a cabbage that produces the most toxic venom, but only for pests. It doesn't affect us. So that's that's a wonder. Then you have modified tomatoes. So I have modified tomatoes because there's a more interesting one coming up after this. So one is spicy tomatoes, which is that scientists have used CRISPR to tweak genes to make them spicy, tweak genes of tomato. So basically how it works is the spice in any fruit or vegetable is caused because of a protein called caspacin. And while studying tomatoes, scientists realized that, hey, look, it has some form of the gene which codes for caspacin. So if I modify it a little bit more, I can get the tomato to actually produce caspacin. And that's exactly what they did. They used CRISPR and they modified the pre-existing kind of similar form of caspacin that already existed in tomato to form the actual gene that codes for caspacin. And now you have spicy tomatoes, and these are very spicy. You can Google it and you'll see people having this tomato and just really burning up. Next is you have anti-cancer purple tomatoes. So first off, these look pretty cool. And so these are purple because they contain genes for anthocyanin. Anthocyanin is a pigment, like chlorophyll is a pigment that makes leaves green. Anthocyanin makes flowers purple. So they took anthocyanin and from one plant, from the snapdragon plant, and put it into tomato. And they did this because they knew that anthocyanin also has antioxidant properties. Antioxidant properties basically also give it anti-cancer properties. So now you have purple colored anti-cancer tomatoes. So, so sometimes, like I've said multiple times actually, that there are mistakes in a gene, and that makes proteins act, the proteins that result don't act the way that they should. So you can use genome editing to correct these mistakes. Like I said, while talking about CRISPR, you can use CRISPR to replace a faulty gene with the right gene. So, okay, so the most expensive medication in the world is a form of gene therapy. So how much do you think it costs? So there will be a link shared on your on the YouTube chat, so you can access it and just take a guess.
Navya, have you shared the link?
Yes, ma'am, I have. It's just the lag between YouTube and Zoom is a little bit, so I think the results.
Oh, yeah, because, yeah, okay, all right. But guesses are coming in, Sandhya. $2.1 million. Some are saying billions, crores,
Okay.
a few lakhs. Someone has written ones with lots of zeros I can't count. Gazillion. Yeah, so children are typing these responses.
Oh, okay. I am not able to see it for some reason.
Yeah, because I think there's a lag in the link coming in the chat and
one that says $1,000 to $100. I think it's a billion.
yeah.
I'm thinking those 2.1 million.
Expecting a lot lower answers because the answer is going to be pretty surprising. It was to me. So it costs it costs 2.1 million. And this is a single dose medication. So you just take it once and that's it. There is no multiple doses, there's no booster dose, there's nothing like that. And it's so expensive because it's gene therapy. So this drug is called Zolgensma, and it's used to treat a disorder known as spinal muscular atrophy. So it's basically they've used a system where they have the corrected sequence, they put that into a virus, and they inject that in you. So the virus goes and infects your cells, and it goes and it replaces the faulty sequence with the right sequence. So that's all the therapy is. But the change that it can bring to our life is people actually, the parents. So there's only one patient who's actually received this drug before, and his parents have to say that it's completely worth it. So yeah. So gene editing tools are used to specifically target these mistakes and replace incorrect or mutated genes with the correct one, like I said. So in the disease that I was talking about, muscular atrophy, so this is your normal looking muscle, this is your atrophied muscle. So from just this one cell, you can see that clearly, this is it's not doing very well. So in this disorder, you have a mutation in this gene, and hence your protein formed is really weird. So your gene therapy is going to go and replace your faulty gene with the right gene. And so yeah. So another application that you have of genome editing is livestock. So a lot of livestock like cows, sheep, even goat are being modified so that their milk is more healthy. So not just healthy, they're using it as a source to get chemicals that are otherwise a lot harder to make. So to get therapeutic components, to get pharmaceutically
The new link that I've sent you, just log in and see if it's working, because some people are saying it's not working. हाँ देखो जरा, मैंने जो उस पे भेजा अपने ग्रुप पे, जरा देखना.
Malabam.
Yeah.
Malaman, please
mute
yourself. Yeah.
Sorry.
Sandhya, continue.
Yeah. So they've modified cows, sheep, goat to get milk that produce the healthier for humans and that also has therapeutic components. That basically milk gives you medication, and you can isolate it from there. And it's a pretty it's easier than synthesizing the medication itself. Chicken are being made to produce more eggs. Pigs are being used for organ transplants because, as you know, we can use pig organs to in humans, like pig hearts and heart valves are being used commonly. So making genetically modified pigs just for this purpose has also been done. And Dolly is the world's first fully cloned animal. And when I say fully cloned, they took a body cell from another sheep and they grew it and they did a whole lot of changes to how it grows and they reprogrammed it. But they got Dolly, a whole sheep that was genetically 100% identical to an already existing sheep. So are all gene changes inherited? So you can have gene therapy earlier in life, or you can have gene therapy, say you're grown up and then you get a disorder. And if that disorder is treated with gene therapy, what's the difference? So the difference is that if you treat an embryo, the changes are inherited. And if you treat humans, like if I inject, say, that drug to your skin cells, then it's not going to be enough to do it once. I'll have to do it over and over because those skin cells are eventually going to die. So what's the difference? So the first one, where you make changes in the embryo is the product of a fertilization of your sperm and your egg. So it's brand new. It's just that single cell is going to give you all the cells of your body. So if I make a change in the DNA of your embryo, that change will be reflected in all cells of your body. But if you're an embryo, you become a person, you grow into an adult, and then I cause a change in your DNA, that change is not going to be reflected all over your body. And that's called somatic cell therapy. So not all gene changes are inherited. But now, considering that there is a possibility, if you use germline therapy, as you see here, you can edit an embryo and the change will be seen throughout the human body. So take everything that I've told you till now, that genes encode all features of a human being. And if you edit genes at the embryo stage, you can get a fully edited human. What does that give you? That gives you the possibility to edit an embryo in terms of what human you want. You're basically putting together different features that you want to create your own baby. So watch this video to get an idea of what I'm talking about.
As scientists perfect techniques of genetic manipulation, especially those of TALENs and CRISPR, the possibilities that become unlocked are theoretically endless. It would become possible to design every part of the genome, whether it's eye color, hair color, disease resistance, or far more advanced traits. What if you could increase brain function, muscle mass, or eyesight? Lifespans could be lengthened, or perhaps new human abilities could be developed. So much so that creating real-life X-Men could come within the realm of possibility.
So someone actually asked the question about X-Men in the flyer that we circulated, and looks like if genetic modification as such goes unregulated, if there's no one to say, hey, look, this is where you draw the line, you can't go beyond this, then it might just be possible. But let's hope we don't get there.
As scientists,
so like I said, designer babies, you design every feature of that particular baby. Is that ethical? Is that right? Do we have the right to actually do that? And that's a question that's being debated very, very strongly, considering that we're entering an era where this might be a possibility. So there's going to be another poll on the YouTube chat, and you can just put in yes or no, or just answer that question there, and we'll get an idea of what your opinions about this are. So this whole ethical discussion about designer babies is a big one, and it's something I think everyone should be informed about. So to talk to you about this whole thing about the concept of designer babies more, and where we're headed as a scientific community in this direction, in this field, what can we expect going forward? And to talk about how genome engineering is not actually it's not that far off. It's not as unreachable as we tend to think it is. That scientists even today are using some form of it in their day-to-day work. I call upon Dr. Koili Mapa, Assistant Professor of Shivnada University. Thank you.
Hello, everyone. Thank you, Sandhya, for a beautiful talk and introduction about the genome editing, genome engineering. So I have very few slides just to give you a visual cue for what I am trying to explain. So let me share my screen. Is it visible?
Yes, ma'am.
Okay. Okay, so Sandra has nicely explained that what is gene and what do they code for. That is majorly coding for some proteins, which are basically the workers in the cells. So they will bring about all the necessary changes that are required to keep us alive. Not only us, all the organisms you see around us. Okay, so starting from simplest bacteria till complicated animals like human beings, all of us are moving, laughing, talking. Everything is possible because some proteins are working in our cells. they're generating energy, they are maintaining the membrane polarization. So everything, so cell division, cell death, everything is maintained, and that's how you are growing from a very small baby to a full adult. So when we know, so people got to know about the structure of our genome and what do they code for. And in the beginning of the century, actually the draft of all genome sequence of human beings were published and were sequenced. So we got to know like how do our genomes code, I mean, how do our genome sequence look like? Like if you can just read the what is there in our DNA, only 4 letters are there, that's true, but my genome sequence will be different from a plant, for example, or a small animal like insect. They are pretty different. Some part of our genome are pretty conserved, which is known as the conservation of the sequence. So there are few part of our genome or DNA which remained conserved over evolution from a primitive organism to the most evolved organism. But major part of the genome got changed, and we have become more and more complicated organisms. So when we study such, let's say, a function of a particular or enzyme, you people are interested about enzymes, and there are several such like thousands different enzymes which is coded by our DNA. Okay, so if I want to compare like a bacterial enzyme which is present, let's say in E. coli, you have heard about E. coli, that's the most common bacteria with which we are working in the laboratory. But this is also present in our gut. Okay, in your tummy it is present, and you may also get infected sometime from nature by E. coli. There are several such bacteria which are present in your gut as well as they are present in nature, and sometimes they may become pathogenic. Pathogenic means they may cause some disease within our body. So if you check the genome sequence, so bacteria also has a genome, they have also their DNA, but this is much smaller compared to the genome what we have, or what an elephant will have, or what a plant will have. Not necessarily a more evolved organism, more intelligent you can say, will have a bigger genome size. Not necessarily like that. A plant genome is much bigger than a human genome. Okay, most of the plant genomes. So there is not such direct correlation, but if you take a much simpler organism like a bacteria or a unicellular organism like yeast, which you find in bread or beer, for example, that contains a much smaller genome. Like how many bases are there in their genome compared to our genome. So using these organisms, you can study nicely what your genome codes for, which type of protein they are forming, and you can compare the 2 or more different proteins of similar properties, let's say from bacteria, from yeast, from other organisms like roundworms, fruit fly, what you can see flying on top of the bananas. Especially you have seen that there are small flies which fly around the bananas if the banana are ripe. And there are several other organisms which are routinely used in the laboratories, which we call them as model organisms. They are beautiful organisms which give us the handle to check and manipulate the genome and see its effect. You cannot always manipulate the genome, cannot ever manipulate the genome of a human being. So that comes under lot of ethical clearance and a lot of like under the purview of the ethical committees all over the world. So it's not permitted yet. But if I want to know, like, okay, we have reported that there is some alterations in the genome that are reported with a group of people who are suffering from albinism, for example. Albinism where the skin color is gone. The skin color is completely white. You might have seen few people who are not having any skin color, okay, or their eyelashes or eyebrows, everything is white, just because the skin pigment is completely absent. The protein which gives this particular skin color, that is faulty or that is not at all working. Okay, so in those conditions, that will be called, not only in human being, albinism is present in many animals. Most of the animals you will find the albinos, which are completely white, colorless. So this happens because of some problems in some of the enzymes which produce the color. Okay, so if I see that a particular problem or particular mutation, which we called as that a ATCG is now changed to ATTG, for example. Okay, and I found a group of people having this particular condition called albinism, having this particular mutation. So how do I prove that? I cannot put this particular mutated DNA in another human body and prove that. I need some organisms where I can check that, and that is the purpose of using model organisms. So here is a list of few of them, and there are many more. So starting from very simple model organisms like a bacteria, which I told, most commonly used are the E. coli or Escherichia coli. This is a prokaryotic organism, okay? So where you do not have a particular nuclear membrane, I think most of you are familiar about prokaryotes and eukaryotes, especially who are in the high school. Okay, so you know that the organisms, how they evolved from the prokaryotes to eukaryotes. In eukaryotes, if you look inside each and every cell, you will find that there are membrane-bound subcellular compartments where most of these enzymes are segregated into, let's say, rooms. Let's say it's a big house where there are individual rooms. One is for watching TV, that's a living room. Another is for dedicated for having your food, that's a dining room. 2-3 rooms for sleeping purpose and all these things. So in eukaryotic cells, which is more complicated compared to the prokaryotes, because they have a different set of enzymes and which are involved in different set of functions, they have been spatially separated into these subcellular compartments. So there are several different eukaryotic species. We all are eukaryotes, and you know that how we have evolved over the evolution. So there are several different model organisms which we use from eukaryotes itself, starting from unicellular eukaryotes. So we all are multicellular. There are so many different cells in our body, but there are organisms, even eukaryotes, which are only having one organism is only one single cell. Okay, so that one example is yeast, which is present in bread, and you can also purchase them from the grocery shop, where you put them in flour along with some sugar and all. Next day or after few hours, you will see the whole dough is fluffy. Why? Because the yeast has grown using this sugar, and because they will produce a lot of carbon dioxide, you will get the bread fluffy or the dough fluffy. So that's the yeast which we routinely use in the lab or laboratory as a model organism, where you can edit the genome and see its effect. Similarly, there are multicellular organisms like roundworms, which we call, the scientific name of which is Hynorbitis elegans. Another very important and very popular model organism is the fruit fly, which I just mentioned, that the small fly which fly around the ripe fruits, especially bananas. You might have seen that if you just keep banana for a few days, after a few days you see that the small flies are flying around. These are nothing but fruit flies, and this is one of the very important model organism what people have used for decades to understand various genetics event and the flow of gene from one generation to another generation, as well as many other events has been studied in these fruit flies. Similarly, so far what I had told about roundworms and fruit flies, among the multicellular eukaryotes or multicellular organisms, these are kind of primitive organisms where they do not have the vertebra and all. But if you move to even the higher organisms like vertebrates, there are several different models like zebrafish. This is a fish model. It allows you to perform several different experiments, especially when fish embryos are developing into fully grown adult fishes. I will come to few of the slides where you can understand what are the applications of these model organisms, especially when you are modifying the genome, for example. And of course, there are vertebrate model organisms like mouse or even primates. Chimpanzees and all are also used as model organisms. So each of them provides you some advantage always also comes with some disadvantage. I am not going too much detail because this may be too advanced for you people. So just to show that, just a second. Yeah, so there are various different model organisms like this. Yeast, I myself work partly on yeast, which are like these unicellular eukaryotes. And most of the molecular biology labs have to use the E. coli bacteria for several different purposes, especially when you are doing molecular biology and genetic engineering. Usually you use some vectors for cloning purpose, for example. There you routinely use this bacteria called E. coli. And these are the other examples which are more complicated, having more and more organ systems, having different types of tissue in their body, like these fruit flies or plants like Arabidopsis, or even the higher ones like zebrafish or mice, which are the vertebrate model system having the vertebra. So this is a nice table summarizing what are the difference between the genome size, like how big their genome size is, or how many base pairs are there present in the genome of these different types of model animals, and whether we have already sequenced the genome or not, like whether we know how the genome is organized, how this ATGC is written in the each and every genome. So these are various different bacteria, archaea, eukaryotes. And here I have shown the one for the human, which is like 3 into 10 to the power 9 approximately base pairs. And we have like almost close to 30,000 proteins. This table is little older, and now we know that there are many more proteins. So you can imagine there are like around 30,000 proteins we are encoding, our genome is coding for, and we are making them. So each 30,000 protein, each of the member has some specific function. And if there is something goes wrong in expressing them, as Sandhya was explaining to you, that a single change in this base pair, as it should be, if it is changed, then the whole protein that it encodes for can change, and it might create a havoc within the cell. Okay, not only the structure changes, its function completely depends on the structure. So each and every protein of any organism you take, they need to be in a particular conformation. We call it in a particular structure. For proteins, this is called the conformation or conformational state. So if you just edit or mutate a particular base in the sequence that is coding for this particular protein structure, you might change the whole protein structure, and in effect the protein function may also change. So as I was giving example of a particular example of albinism, not only that, there are several different such genetic disorders you might have heard or read, where a single mutation has caused the problematic proteins, which is making it incapable of its function, ultimately causing some disease, for example. So how to prove that this kind of mutations are causing actually, or the causative factor for such disease? So as Sandhya was telling that indeed you can change these genomes of organisms, and this is a very popular model organism, especially for scientists who understand the development from embryo to a adult organism. Okay, so this is a very popular model organism just because of the fact that the embryo is completely transparent. You can look through, you can put it under the microscope, you can see the events of cell division and formation of the organs. So isn't it beautiful? So that you can just put a fish embryo and just can watch over time for a few days and then figure out like how the egg or embryo is transformed into an adult fish. So that can be visualized under the microscope because the embryo is transparent. So that's why this particular model system is a very nice and very popular model organism. Usually the adult fish has having the stripe structure that's like a zebra. That's why these are known as the zebrafish. So what scientists have done, that there are many more genetically modified zebrafishes which scientists have created, Like, for example, this king bow fish, which is expressing many different colored proteins. Basically, these are different fluorescent proteins which has been engineered in its genome, and the fish's skin can look like a rainbow. That's why it has been named as a skin bow. Actually, previous to this particular fish, there was a report called brain bow. Brain bow means the brain cells or neurons were expressing different colored proteins and looks like a rainbow. So from this, the concept of this rainbow followed by this particular fish called skin bow came, where you can express multiple different proteins of different colors. And there are examples, what Sandhya has also shown, that you can make beautifully colored fishes, you can keep them in your aquarium and enjoy. But is it the only purpose of engineering such genome of these model organisms? Not really. You can do it for fun, but you know that Sandhya has given you a glimpse of how expensive the process of genome editing or genome engineering can be. So in model organism level, it may not be that expensive, but it's certainly quite costly affair. So no one will do it just for fun. So indeed, we are doing, or scientists are using genome editing or genome engineering to understand the biology. For example, in this particular example, here, this GFP protein, which is originally obtained from the jellyfish, has been, the DNA coding for the GFP has been introduced into a particular gene, which is specifically present in the vascular system. What is vascular system? All the blood vessels and heart. So in the wild type fish means which has not been edited or which doesn't has any mutation. There we see that all the blood vessels are how nicely they are distributed all over the body, both on the dorsal side towards the back as well as the ventral side. But if there is a mutation in this particular gene, we see the formation of these blood vessels are quite disturbed. So this is a mutant fish where we see the ventral side vasculature is completely deformed, and there is another mutation, let's say, where it is completely deformed and there is no such vasculature in the dorsal as well as on the ventral side of the fish. So how will this fish develop into adult? Most probably, if you follow it over some period, within 2-3 days, you will see that these embryos carrying this particular mutation will die, whereas they will be making for a few more days. And these are wild type fishes, like they don't have this particular mutation, they will be developing into the adult fishes. So that's how over or during development you can use this transgenic. So this is a transgene which is not the original gene present in the fish, but you have taken it from some other organism for your purpose and made a transgenic. So in case of fishes, these are known as the transgenic lines. So these transgenic lines can tell you or can answer, if I have this particular mutations in this particular gene, what will be the effect? So you can make a kind of inference. Okay, this is required, this particular mutation is actually detrimental or problematic during the development. So people who are carrying this gene, most probably they will have, their babies will have problem in the formation of this blood vasculature, and most probably they will not survive into the adult life. Okay, so not necessarily always you can the same mutation will have the same impact across all organisms, but in most of the cases this is true. So that's why we use the model organism. So this is another evidence of modified or transgenic animal. This is another vertebrate model system, mouse. Mouse, you know, doesn't express GFP, which is green fluorescent protein. You have never seen a green mouse, isn't it? But this mouse has been genetically modified, where the GFP protein has been introduced into its genome, and now you see that most of the organs, so this is the digestive system, starting from the tongue till the anus, you can see it's a completely green. The whole mouse is actually green. So nobody has done this for fun. So there was a purpose. They needed to understand that how the organs are developed, and if I have some particularly on this particular picture, you can see there are some red dots. So these red dots are actually the human tumors cells which has been transplanted in the mouse and has been observed over time how this grows. And of course, if you manipulate other gene, you can see the effect. Maybe manipulation of the other gene will make the tumor bigger. Okay, so how do you manipulate? You have seen that human or females who are suffering from the breast cancer is having a particular mutation in a particular gene, and that particular gene has been mutated in this model mouse, and along with that, the human breast tumor cells have been implanted in the mouse to see whether, compared to a wild type mouse where the mutation has not been introduced, whether this tumor is getting bigger or in more rapid manner it increasing. So that gives you an idea whether the second gene which has been mutated or the gene which has been mutated in the mouse, which we have idea from the human real life example, whether that is causing an effect on the breast tumor progression or not. So all this gives you an idea. Yes, you can change the genome, you can edit the genome. Okay, so there is a particular writing in our genome, ATCG and all, which if it is faulty, then you can artificially remove it, correct it. Or I can also introduce some extra sequence or a foreign sequence to make a transgene or transgenic animals. So these are the commonly used tools for genome editing process, and Sandhya has already explained a little bit about the most recently discovered one, CRISPR-Cas9 system, for which Nobel Prize was awarded in the last year only. Not only that, there are some engineered genome editing tools as well. What do I mean by engineered? At least the Cas9 or CRISPR-Cas9 system has been obtained from nature. We know from some bacteria contains it, and we have applied it for our purpose. These are engineered ones where this has been, this is basically an artificial molecule where some of the proteins which has a DNA binding property with particular sequence has been, again using the genetic engineering knowledge, has been coupled with a endonuclease which can cut the DNA sequence. So basically this part of the tool is to figure out or to target this particular tool to a particular, because you can imagine like we have like 3 into 10 to the power base pairs. How do we know that where exactly it should be cut and edited? So for that you need to give it a specificity which is obtained by this part by specific binding property of these modules or proteins, which are in this particular cases these zinc finger nucleases or these talents. In case of talents, these are the modulatory proteins which can give a specific binding to the genome. So you can define the specificity where it should be changed. And there are repair processes because you are cutting the genome, you are cutting the double strand. There are some cellular pathways by which it will be corrected and again joined back. So the problem is like whether we are always applying it to understand some basic biology or only for good. So this is a recent controversy in the field of genome editing of human. Okay, so this report came in not so old report, only 2 years back, little more than 2 years. In November 2018, this scientist from China, He Jiankui, who actually he didn't disclaim, give announce this particular discovery, but another report came where they reported that this particular group from China has actually edited the human embryo, genome of the human embryo, and they have given birth to actually genome edited human babies. So followed by this announcement, he actually was forced to reveal that yes, indeed, he had done some genome engineering of human embryo, after which twin babies were born. And he claimed that he did it for good. Okay, so what did he do? So he has disabled a particular gene which is giving rise to or encoding for a particular protein called CCR5, which is used by human immunodeficiency virus or HIV virus, which causes the disease called AIDS. All of you have heard acquired immunodeficiency syndrome. It's almost a deadly virus if not treated in time. Okay, so our immune system gets weakened over time, and then ultimately the person will die from several different infections because the immune system is completely compromised after infection. The human embryos, and then the purpose was to make the children who will be born from these edited embryos to protect them from the HIV infection. Okay, so why did he do that? He was actually running a in vitro fertility clinic where he was mainly trying to help the couples where mainly the fathers were infected with HIV and the babies were having quite a lot of chance to have HIV infection during or after birth. So he claimed that the purpose of genome editing or genetic engineering on the embryo was to protect the babies from the future HIV infection and save them from the probable AIDS. But this is a concern whether we will be now heading towards the designer babies, like I want my baby to be fair and tall and beautiful, whether we should be going for that. So that's a controversy, and of course we want to know what is your idea after hearing this, that whether we should be going for the designer babies or not. So still it is under ethical, no one has given ethical clearance to edit the genome of the human babies at least, and for most of the animals also we require ethical clearance and seeing the risk associated with it. Okay, because whenever you are doing the genome editing, not necessarily only one site where we have targeted the DNA will be modified. There are most of the cases the other part of the genome might also get modified. Okay, so the risk is always there. So now the forum is open for your questions and discussion, and I am open for any questions that you might have. Any question?
Can I ask a question?
Sure, my sure.
This is such an enriching session, and time is less. There are many doubts which are coming to my mind because the time was only 1 hour. Probably we'll have, a list of questions which are coming. We'll mail to you also.
Sure. That will be really nice.
Would like to hand over to Sandhya and you now. So Sandhya, please carry forward.
Thank you, ma'am. Thank you so much for that. So Gargi is up next, actually. So, yeah.
Yeah, it was indeed enriching. And now I welcome Satvik. Satvik Das is a student of grade 9th from Shivnada School, Noida. He developed a nuclear website. Satvik, please may I request you to take over the further proceedings.
Yep. Thank you, Gargi. I am now going to present my screen to give everyone a tour of our Nucleus Science website. So, after the major success of our inter-campus mega event, the Energy Boot Camp, it seemed only befitting to launch our inter-campus science website, Nucleus. By launching this website, we wish to maximize the outreach of the students and faculty of Shivnada School, Shivnada University, and the Shivnada Foundation in this digital era. Without further ado, I introduce you to our website, Nucleus. Our first page aboard to getting to know us is our homepage, where we brief a visitor about our vision and our journey. The workshops or live classes are where one shall find the resources for workshop training and other important links, such as this very session. In the news is the page that sums up all the current affairs relating to many interesting events that are open to attend, such as today's event on Crispr, the gene editing tool, with our senior mentor, Ms. Sandhya Kasi from SNU. The resources page is your one stop for compilation for every resource for all students alike.
Can you show the team thing so that Dr. Mapa can also see that there are university professors?
Our magazine page is the page that contains our monthly newsletter that keeps one up to date with all the current happenings. And finally, our team page, which is a homage to everyone who put in a lot of effort towards making this initiative come to life. This is the most important page containing the faculty from all 6 campuses and our mentors from Shivnada University, with all email IDs mentioned, following our motive of inter-campus relations. I would also like to thank Ms. Sonia Sharma from Shivnada School, Noida for guiding us through this website. And that is the tour of our Science Nucleus website.
Thank you, Satvik.
Nice.
This has indeed been a very fruitful session. I would now like to invite Ruchi ma'am to give the vote of thanks.
Thank you, Navya. Good evening, everyone. Joining me in extending the vote of thanks today is Ms. Mala from Shivnada School, Noida. We, on behalf of Team Nucleus, want to thank Dr. Mapa for sharing her work and opinions with us. Dr. Mapa, we are delighted to have you with us today. Thank you so much for the informative session and for sharing your insights and expertise.
Thank you for the invitation.
Yes, thank you so much. A big thank you to Ms. Sandhya for explaining so well to all of us what genome editing is. Your content was very engaging and quite informative. I'm sure our audience feels the same way. So thank you both of you for enlightening us with this very interesting topic. A sincere thanks to each and every one of the audience who are present today and for making this session fruitful. I would also like to extend my thanks, please excuse me, to our student mentors, the moderators for today, Navya, Arushi, as well as Gargi. Thank you, Satvik, for sharing the Nucleus website with us. I'd also like to express my gratitude to the IT team for their back-end support. And lastly, thank you, Vinita ma'am, Gulfam sir, and all the student and teacher mentors behind Team Nucleus. With everyone's collaborative effort, this event came to fruition. Looking forward to having more such sessions in the future. Thank you so much.
Thank you. It was such a wonderful session, and thanks to all, and thanks to Matthew, who is always there with us.